Hello viewers, welcome to CEC-UGC Gurukul lectures. I am Dr. Soni Rastogi from the Chemistry Department of Sri Aurobindo College, University of Delhi. Today I will be giving a lecture on pharmaceutical chemistry. The topic of my lecture is drug discovery, drug development and expensive failures. This is the first lecture of the series. In this lecture, I will talk about the important stages in drug discovery and drug development. It will begin with the choice of the disease, choice of a target molecule, target specificity and selectivity, and end the lecture with the test system. This is the topic of my lecture, part 1. Drug discovery, drug development and expensive failures, part 1. This is the part 1 of the series. We will start with the introduction first. Introduction gives a brief description of drug discovery and development. In earlier times, medicine consisted of herbs. The natural products obtained from plants were used as therapeutic drugs to cure the various diseases. Therapeutic drugs means the drugs which are used to cure the ailments and give you the relief from the pain. Pharmacologists made synthetic analogues of the natural products which gave way to drug discovery and drug development. Development in the field of medicine and advancements in biosciences help the scientists to understand the functions of the human body at molecular level. This led the pharmaceutical industry to design a therapeutic drug which can interact with the target or receptor with high binding capacity, resulting in minimum side effects. So, in drug discovery and drug development, better is the drug if the drug target interaction is maximum, and then there will be least side effects. During drug design, it is a crucial stage in drug discovery and drug development. It is the method of designing a medication in a manner that there is maximum interaction between the drug and the target with least side effects. This is the basic concept of drug discovery and development, that when we design the drug target interaction has to be minimum so that there is least side effects. Drug design is the innovative method of discovering new medicines based on the knowledge of a target. Drug design involves the designing of molecules in such a manner that it complements the shape and change of the target with which it interacts and binds. In today's times, there is frequent reliance on computer modeling techniques and bioinformatics approaches. The ways of designing the drug includes ligand-based drug design, structure based drug design, and computer aided drug design, that is called CAD. Apart from this, there exist multiple strategies to improve the interaction between the drug and the respective target or the receptor molecule where the enzymes are present. Earlier, herbs were used to cure diseases. In 19th century, the concept of drug discovery and drug development was evolved. Drug development is the method of introduction of a brand new drug into the market once the lead compounds are identified through the method of drug discovery. The lecture will begin with briefly introducing the process of drug discovery and drug development. It will describe the various stages involved in the process, beginning from choosing the disease to clinical trials. It was in mid-19th century that serious efforts were made to isolate and purify the active compounds called lead compounds from these natural products. Scientists synthetically made drugs similar to herbs for curing lives of millions of people. Chemists made synthetic efforts to make thousands of analogues similar to what nature had provided. The recent advances in medicinal chemistry and better understanding of the functioning of the body at cellular and molecular level gave a boost to the research being carried out in the pharmaceutical industry. The research begins with the identification of a target molecule and designing a drug competent to interact with that target, resulting in minimum side effects. So, in all, we can say that the gist of drug discovery and drug development, or when we design the drug, is that there should be maximum interaction between the drug and the enzymes present in the target to give better efficacy of the drug and minimum side effects. While increasing research and development, various advancements involving of genomics, study of genes, comprehension of the disease at molecular level, and easy and convenient availability of data have given way to the expansion in drug discovery and development. Genomics is the study of genes, differs from with genetics, as it aims at the collective characterization and quantification of the genes. And genetics, in genetics we talk about the individual genes and not of in quantification of the genes as we talk in genomics. I will be using certain terms quite often in my lecture, like lead compound, drug and target molecule. I would like to give brief introduction to these terms because I will be using them quite often in my lecture. There is not much difference between the lead compound and drug. The compound undergoing the clinical trials to be transformed into drugs is referred to as the lead compound. On the other hand, the drugs are compounds that come into the market and are prescribed by the doctors after undergoing clinical trials. In simple words, the compound is called lead compound during the process of clinical trials, and the final product after the clinical trials is called the drug. Then we will come to the target molecule, and again I will be using target molecule or the receptor molecule quite often in my lecture. So I would like to just get you aware with the term target molecule. Target molecules are the tissues or the organs present in the body where the disease is located. The enzymes are present in the target. When the drug enters the body, it interacts with the enzymes present in the target molecule and results in drug target interaction. More the drug target interaction, better is the drug and lesser are the side effects. These are the important points in the drug target interaction and in drug discovery and drug development, or when we design the drug, that drug, more the drug target interaction, better is the drug and lesser are the side effects. This is the basic concept of drug discovery and drug development. Now we'll come to the stages in drug discovery and drug development. The first stage in drug discovery and drug development is choose a disease. The process begins with identification of a disease by a pharmaceutical company for which there is demand for a new drug. The companies are driven by the motive of good financial returns for their investment. As a result, the research projects for the designing of a new drug is carried out on diseases such as migraine, depression, obesity, flu, cancer and cardiovascular diseases, as these are more common diseases with high demand for drugs instead of tropical diseases with low demand for drugs. In other words, we can say that research and development carried out by the pharmaceutical companies on common diseases, which is common to most of the population of the country, like depression, obesity and cancer, where there is a demand for drugs, rather than uncommon disease like elephantitis with a low demand for drug. Elephantitis is a very severe disease though, but it is very common in some parts of Africa. So the demand for that particular drug is very less, but the demand on the other hand for certain common diseases like depression, obesity and cancer is there. So there is a demand for that particular drugs. Now, the second stage in drug discovery and drug development is choosing a drug target. Once a particular disease has been determined, a suitable drug target, that is the receptor or the enzyme, has to be identified. This is very important step as drug reacts with the enzymes involved. Therefore, before talking of drug target, we should know the molecular structure of the target with which it would react. An example of developed drug targets are caspases, which are enzymes present in our body. Caspases act as catalyst in the process of hydrolysis of cellular protein. The product form results in inflammation and cell death. We know cell death is a natural phenomenon taking place in our body. Without these enzymes, there would be unregulated growth resulting in disease like cancer. Further, this property of caspases, which is an enzyme present in our body which catalyzes the hydrolysis of the proteins, can also be used by the pharmacologist to produce new therapies for a variety of diseases requiring cell death like cancer and certain viral infections. So, at one hand we can say that caspases is an enzyme which is present in our body which is doing the inflammation and cell death, but at the same time that property of caspases can be used to cure dreadful diseases like cancer, where you need cell death, cell breaking of cells. The third stage in drug discovery and drug development is the target specificity and selectivity between species. The more selective the drug for the target, more likely that it will interact with the specific target and less chance that it will give undesired side effects. The best targets are those which are unique to the disease causing microbes. Target specificity and selectivity is also essential for drugs interacting with their own enzymes. Few targets acting as enzymes, inhibitors, should be selective in nature, ensuring that they only inhibit the activity of target enzymes and not some other enzymes. We know that targets are very specific in nature and selective in their nature. So we have in the drug design that one should ensure that they only inhibit the activity of the enzymes present in the particular target and not some other enzymes. So this we can say that target specificity and selectivity of the in drug design and drug development. Targets are enzymes and we know enzymes are specific in nature. The drug interacts with specific target, ensuring maximum drug target interaction and reduced chances of undesired side effects. If we take the drug, for example, penicillin, into consideration, it only targets the enzymes which is involved in the biosynthesis of bacterial cell wall. The role of it is that it inhibits the synthesis of bacteria cell walls. However, the cells of mammals like human beings do not have cell walls, so this enzyme is absent in humans, and thus the drug penicillin acts only on these bacteria and has no side effects on humans. That is, they work only on the bacteria, kill them because they inhibits the bacterial cell wall synthesis, and the humans are free from that disease as they have no cell walls and there is no effect on humans. The fourth stage in drug discovery and drug development is choice of test system, or we also call it bioassay. Human testing cannot be done at an early stage, and so the test must be done in vitro, that is isolated cells and tissues, or in vivo on animals. In vivo, that is in animals, the animal is treated and is under observation to see whether the drug eliminates the observable system, which means that drug is able to eliminate the particular disease. Such testing is very slow and causes animal suffering. The tests which are done in vivo on animals, they are very slow because we have to observe the animals which are undergoing this testing for years together, and at the same time they cause animal suffering also. If a negative result is observed, then it is difficult to tell whether it is due to the drug failing to bind with the target or the drug not reaching the target at all. We know that when a patient takes the drug, it has to travel through the body, crosses number of barriers, interact with the enzymes present in the target molecule, but number of times it happens that drug fails to bind with the target, or the inefficiency of the target can be also that the drug does not reach the target at all, because when it travels through the body it has to cross number of barriers. Number of times many drugs which proved effective in animal testing were ineffective in clinical trials. For example, this is a very good example. We can quote that the drug which was found very effective in clinical trials, it passed all the tests of clinical trials, but when it was done on the human beings, it didn't work. Like penicillin methyl ester are hydrolyzed in the mouse or rat to produce penicillin, but not hydrolyzed in rabbit, dog or man, that is human beings or mammals. The fourth stage is screening under nuclear magnetic resonance. Nuclear magnetic resonance (NMR) spectroscopy can be used to detect whether a compound binds to an enzyme or receptor target. In NMR spectroscopy, when the nuclei relax to the ground state, from the excited state it gives off energy. This energy is used to produce a spectrum. And we know in NMR spectroscopy the time taken by different nuclei to relax from the excited state to the ground state is called the relaxation time. Small molecules such as drugs have long relaxation time, whereas large molecules have small relaxation time, and thus the measurement of small molecules is only possible. If the drug fails to bind with the protein, then its NMR spectrum is still detected, but if the drug binds, it becomes a part of the protein, and as a result the nuclei will have a short relaxation time and no NMR spectrum will be detected. So NMR spectrum is not detected for the drug target because it becomes the big molecule. Once the target and the testing system have been chosen, the next stage is to find a lead compound which shows the desired pharmaceutical activity. Many natural products are biological active compounds and are used as lead compounds. Example: morphine, cocaine, quinine. Useful drugs which have been isolated and identified are Taxol from the yew tree and anti-malarial agent artemisinin from a Chinese plant. Taxol is an anti-cancer drug which is used in chemotherapy in patients suffering from cancer. So these are the two structures of artemisinin and Taxol. With these two structures, artemisinin is an anti-malarial drug and Taxol is an anti-cancer drug. Both of them are products which are obtained from natural products which are obtained from the plant, and they have got number of binding sites which are used for the interaction with the target molecule. Artemisinin, an anti-malarial agent, and Taxol, an anti-cancer drug, both are natural products obtained from plants. And as I have told you that artemisinin has got number of potential sites like hydroxyl, methyl. We can replace these binding sites by other groups and study the biological activity. Similarly, Taxol also has potential sites like hydroxyl, carbonyl, methyl and acetoxyl. We can replace these binding sites with ether groups and study the biological activity. The study of biological activity of the natural products can lead to the formation of the lead compound. It is these biological active sites that interact with the target molecule. So many drugs are obtained from marine sources like corals and sponges, as they have chemicals with anti-inflammatory and anti-cancer characteristics, and certain venoms and toxins are also obtained, and in this the compounds which have their structure determination can be done with NMR, infrared spectroscopy or crystallography. And the next aim is that we study the biological active sites of the target molecule. So this is again a natural product called glipine, which has got number of active sites which have been encircled like hydroxyl, and these we can study the active sites of this glipine and study their biological activity and can be used as the drug. Glipine is again a natural product and has a number of potential active sites. Now this shows the drug target interaction with the diagram, and this interaction is between the drug and the receptor or the target molecule, and the explanation of this is given in figure 1. If we see, the proton of the hydroxyl group of the drug is involved in hydrogen bonding with the receptor. If the proton is replaced by the methyl group, then the hydrogen bond is lost. So we are talking of drug target interaction with the hydrogen bond. But in the figure 2, the oxygen atom of the drug forms a hydrogen bond with the amino acid residue. On replacing the proton by the methyl group, the oxygen is still present in ether analog of the drug, and now the bonding is between oxygen and ether, and the hydrogen bonding is weak due to shielding by the methyl group. So in conclusion, we will conclude that drug design is a complex process involving various steps. It involves several continuous and challenging experiments. This method involves each steps are laborious and time consuming, resulting in many years before the drug is marketed. Thank you.